Hello, and welcome to Let's Meet the Virologist, a podcast about the people behind today's virology headlines, people working to understand viruses and how they affect you. We are talking with students, postdocs, and other virologists so that you can learn who they are and what they do. I am Larissa Thackeray, and I am hosting this podcast from America's Heartland in St. Louis, Missouri. On June 3rd, 2022, we talked with Ashish Srivastava, a faculty member at Amity Institute of Virology and Immunology in India. His lab develops highly sensitive CRISPR-Cas-based detection methods for known plant viruses such as begamoviruses and potyviruses. He is currently learning how to analyze data from nanopore sequencing to identify plant viruses and hopes to bring this technique back to his lab in India in order to identify novel plant viruses affecting crops. Thank you for talking with us today. Why don't you tell us a little bit about yourself?
Hi, thank you for giving me the opportunity. Hi, I'm Dr. Ashish Srivastava. By passion, I'm a scientist, but by profession, I'm a faculty at Amity Institute of Virology and Immunology at Amity University, India. And presently, I just came as to learn some techniques and more research. I came University of Arkansas at Fayetteville under Professor Yanis Xenetikas.
Great. And can you tell us sort of the path that you followed in your education? Like, how did you get to sort of through undergrad? How did you first become interested in science? Things like that.
Okay, so actually, I belong to a very small town in India. It's in Lalitpur. So my initial education was going through that place. My father was an administrative officer and my mother was a teacher. So I always having the interest in both the jobs. So initially schooling has been done from that place. But because that town is very small and do not having the place for higher education, so I just planned for some examinations. I qualified that and joined Rasan Tukrodi Maharaj Nagpur University for my master's in biotechnology. So the biotechnology is a very broad subject and it is having many aspects to learn: microbiology, medical microbiology, biostats, biochemistry, molecular biology, and many more. So it gives a great opportunity to learn and understand all the subjects and make your preferences. For master project, I joined Dr. S.K. Rath's lab at National Botanical Research Institute, Lucknow, and that helped me to make my mind for plant virology research. So after qualification of CSR National Eligibility Test in India, I preferred to join his lab and completed my PhD from there. I just learned several techniques like scanning electron microscope, transmission electron microscope to study the virus and the host symptoms, and the PCR, ELISA techniques for the detections, and several bioinformatics techniques. After finishing of my PhD, I just planned for the postdocs. I got few fellowships to work in India also and for outside. But I prefer to join the University of Edinburgh at UK. And I just changed my subject to mycology because at that time I was having some instability in my mind, which route should I choose? So I chose in the mycology field, and I worked with Professor Gary Locke. But later on, I realized that I'm having more interest in the plant virology. So I just moved back to India and joined Amity University as a faculty. In this time, I received 3 research grants to establish my own lab. So with the help of our head, Professor Narayan Nishi, and the support of my 3 more faculties, the Dr. Vanita, Dr. Nandlal, and Dr. Sunny Dhir, who got the research projects at the same time. So we together made a lab, established a lab at Amity University, and hired our PhD students, trained them, and now we are having a good facility at there. And post-COVID, I feel that I should also learn some more techniques to bring in our lab. So I moved to US at University of Arkansas to learn more about that.
So can you tell us a little bit about the plant virology that you have been doing in your lab? Sort of what's the bigger picture, and then maybe what are some of the techniques that you use to study that particular virus or that area?
So I just would like to start from my beginning.
Mm-hmm.
So because I'm working in plant virology from my master's, our basic work was to characterize or identify the viruses present in the crops in India. Because in India are having the major issues of bigmo viruses and potyviruses in the crops. So I just started to learn the techniques for initial detection, and then the complete genome characterization. So for bigmo viruses, I identified 3 new species of the virus infecting Jatropha, the Jatropha leaf mosaic Tanya virus, Jatropha mosaic Lucknow virus. After that, because I'm also working on the potyviruses, so I also learned the techniques to characterize the RNA viruses. Beyond the identification of new species, I was also working for identification of novel host for the viruses. I worked to find the viruses from opium poppy, amaranth, moonbean, jatropha. So many crops I have used for that purpose. So after finishing of my PhD, at that time there was a technique emerging named the CRISPR-Cas. So I focused on that work. So when I back to India, I write the research grants and received 3 projects on that aspect in the next 3 years. I established the CRISPR-Cas technique for the identification of bigmo virus in the lab, and we proved that this technique is more sensitive than the PCR. Later on, I also received a major grant for utilization of CRISPR-Cas technique to deliver in the plants using the virus genome. So in this project, we are trying to clone the whole CRISPR-Cas cassette in the genome of virus and trying to deliver in the plants. So we were having good results with that, but actually our work has been affected by the COVID. So yeah, definitely we are behind the time to deliver the goals, but I hope by the end of this year, I will having better results that could be published.
And this is the idea of sort of almost like using the CRISPR-Cas as a way to do gene therapy in plants. Is that the idea?
Yes. So the same thing that has been done with the humans using the AAV vectors.
Okay. So could you talk a little bit more about that? So I think a lot of people, and myself included, have heard about sort of using CRISPR-Cas or gene therapy in the context of human disease, but they may be less familiar with the whole idea in trying to combat sort of pathogens of plants. So could you talk about that a little bit more?
Yeah. So the project that I'm handling currently is deals with the use of virus genome for the CRISPR-Cas, because the making of transgenic in the lab is actually time taking, even if we are working in the lab host, which is having very small lifespan. So my idea is, suppose if you feel that some particular gene helps, a particular gene is helping to support the virus life. So if you are able to target that gene with the help of a CRISPR-Cas system, you can actually will be able to study the role of that gene in very less time. So that was the overall idea, to utilize the plant genome for delivering of the Cas9 in transient way.
And would it be able to be used also, though, as a therapeutic? So if you have crops that are being affected by this particular virus, could you use it then as a way to try and minimize the effect of that virus on the plant yield or something like that?
Ultimately, viruses actually hamper the yield of the plant. And at the moment, the recent project that we are doing in our lab is to develop the CRISPR-Cas13 technique to induce the resistance against the plant RNA viruses. This technique is at present, we are performing in Nicotiana benthamiana plants. So we have designed some constructs and establish the expression of Cas13 protein. So we are having the Cas13. We have designed the guide RNAs and we are utilizing them to target the RNA viruses in vitro. But later on, when we will be successful in that and we find the suitable RNA CRISPR RNAs to target that region, we will translate the whole system first in the lab host and later on in the main host. So this is our plan for the future.
And then can you tell us a little bit then about this work that you're now doing in the US to sort of add to your, it sounds like repertoire of skill?
So in USA, because I was not having much experience with the high throughput sequencing techniques, so I joined this lab, and here I got opportunity to learn more about a technique, MININE of Oxford Nanopore technologies. So this is a very small device that you can put in your pocket and just insert in your normal laptop and analyze the data. So here what we are doing, we just took infected plant, extract a whole nucleic acid, and after all the processes, we just put it in the device, and we are able to identify the unknown viruses residing in that. So this technique is really very good for me, and I'm still learning the analysis of the data, how we will perform, we are still trying to analyze the data to find the more and more viruses which cannot be detected through PCR.
Great. And can you describe or talk a little bit more about Nanopore? So people have been talking about this for years, and that, it's cheaper potentially. It has, I believe it's like generates longer reads, so that there's, more information that you can get. Can you talk a little bit about how Nanopore is beneficial maybe in resource poor areas or in other ways compared to sort of the more traditional next gen sequencing that people have been doing?
So if you will talk about the perennial crops, they do have the viruses for years, but we could not identify. And if you are planning to do so, you would have to go for the next generation sequencing, which is actually the costly for us also. And also it takes time. So at least, the technique is really good. But if you will talk about for virologist perspectives, our major objective is to find what are the viruses present in that crop. This technique is really help us to identify, even if you are getting a very small transcripts of the virus, you will have an idea that it is present in that, and you can just design your primers and try to pull out the whole genome from that. So it is really helpful for us.
Great. And I'm not as familiar, honestly, with sort of the diversity of plant viruses. So we do some studies with sort of collaborators that study the mammalian virome, as it were. Can you tell us a little bit about sort of the virome of plants? How diverse is it? How complex is it?
So plant virome is really very diverse. If you will talk about perennial plants, they do have several viruses, and surprisingly, there are many viruses that actually cannot cause any symptoms, but later on, if they get a supporter virus, they both just put together their proteins together, try to suppress the immunity of virus and infects it well. So there is no limit. Many viruses can be present on a plant. It can be transmitted through the vectors. It can be transmitted from plants to next generations. So the virus is there can be many plants which looks asymptomatic, but they contain a good amount of viruses in them.
And is there in plants the idea that some viruses are actually beneficial? So in like mammalian hosts, you always talk about sometimes there's benefit and to some viruses, especially asymptomatic viruses, that they may be priming, immunity or something like that. Is there some thought that that might be happening in plants too?
I don't think so. That viruses are beneficial for the plants. Yeah, sometimes if they are suppressed They can help in boosting the immunity by providing some siRNA resistance, the siRNA based resistance in the plants. But most of the time, plant viruses are not beneficial for the plants.
What are your plans next then? What is sort of like your short-term, midterm goals going forward?
So my plan is to explore more the technology CRISPR-Cas. I am still working hard to develop the resistance in the plants using this technique, and also I'm trying to develop novel methods to detect the viruses using this technique. Recent paper I published in Journal of Virological Methods, where I tried to explain whether if we are not having the probes to detect the virus, how we can utilize the alternative dyes to detect the viruses. So my research will be based on the CRISPR-Cas technology, and also my major target will always be to detect the new viruses residing in the field, and characterization and development of their infectious clones to prove whether they are having major impact on the crop or not.
And are you planning to go back to India to sort of pursue this work, or what are your plans?
Yeah, so I'm always having plan to go back to India, because I'm having 2 PhD students under my supervision who is completing their work. And also I'm having a faculty position, and I have established a lab. So definitely I would like to back to India after learning all these techniques, and will establish everything there.
Great. And are you presenting to this work at ASV this year or no?
So I'm having planning to demonstrate whole of my research journey, which is based on the virus detection, characterization, and utilization of CRISPR-Cas technology for their diagnostics.
All right. Well, thanks so much for talking with us today, and we look forward to hearing more about your work.
Thank you so much.
This has been Let's Meet the Virologist, a podcast about people who study viruses. This is your host, Larissa Thackeray, and thanks for listening. You can find us on Google, Apple, Amazon Music, and other podcast providers, or at lmtv.podbean.com.